Hello, this is Martin Wilson welcoming you to this Bite-Sized Bio web seminar, which today is sponsored by Arrivas. Arrivas is a market-leading software company focused in life sciences. With a broad product portfolio, their solutions address both industry and academic environments. Arrivas offers software solutions for the interactive visualization and analysis, especially of very large image data in microscopy. Adaptable, forward-thinking, collaborative, and easy-to-handle solutions are foci of Arrivas's future-facing development goals. Today's presentation is titled Your Guide to Complete and Fast 3D Image Analysis in Microscopy and is being presented by Dr. Chris Zugates from Arrivas. Chris Zugates is an application engineer at Arrivas. He started in imaging 20 years ago as an application scientist and project manager in Raman-based imaging systems, where he took a particular interest in Raman-SEM correlation. Then, after training as a developmental neurogeneticist, he ran one of the first truly industrial-scale imaging projects in modern brain science. He appreciates the difficulties of imaging at scale and endeavours to help Arrivas's customers get the best possible results from massive, challenging image sets. As always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Chris at the end. The recording of the webinar will be available at bit.ly/3D image analysis webinar, all one word and lowercase. So now, over to you, Chris, for the presentation.
Hi, everybody. This is Chris. When you say that I appreciate the problems of image analysis, I'll phrase that another way. I feel all of your pain. Today, we're going to present to you a guide to complete and fast 3D analysis in microscopy. And as many of you know from working with imagery, something like a complete guide to analysis is a bit ambitious for a 1-hour presentation. But I think today what we'll endeavor to do is take a few steps together forward, and I'll show you some of the tips and tricks that I use for performing 3D image analysis, and hopefully in the future we can take more steps together. So the first thing I want to say is image analysis is everywhere. It's something we do all the time. In fact, I just did it 30 minutes ago. I went downstairs to have a bit of a snack, and in the refrigerator there are two kinds of yogurt. There's this really sugary yogurt that my wife likes, and there's this really good Greek yogurt that I like. And of course, I instantly can recognize visually my kind of yogurt, and I can pick it up immediately. And it's this kind of categorization and understanding of things in the visual space that we're endeavoring to do in science with our data. So image analysis, it helps us gain insights about the scientific materials that we work with, and image analysis software is pretty special. It enables us to record not only these data, but it enables us to record the things that we find in the data. And it also helps us to focus in on specific objects and maybe even ponder them for some time. So you may want to study some object in your image. Maybe one day you don't have an idea about what this is or how you could analyze it, but maybe on another day you do. And software gives you the ability to hold on to it, come back to it another day. It also gives you a way to easily compare mathematically, algorithmically, these different kinds of objects that you see in the image. And then, of course, as scientists, what we want to do is we want to make abstractions about these objects, come up with general principles and descriptions about these objects and standard ways of viewing them, and come up with new theories. So our visual system and brain is really good at at parsing out all of this information around us. And nowadays we have instrumentation that is helping us to see even more about the world around us. And this is in the form of very high resolution imaging systems that capture the tiniest of objects, the fastest moving objects. And now we'll be able to see things that we could never before see. I really like to go to this example in the history of science. I think we can learn so much about what we do from the Copernican revolution, and I think it applies in the realm of software, how software can help us as well. So very briefly, we have this guy Tycho Brahe, who builds really the best instruments of the time for collecting the most accurate measurements in the heavens at the time. And this is like what the imaging system manufacturers are doing for us these days. They're building these amazing instruments. And also we're having these wonderful advances in histology and tissue clearing. And so now we can apply the latest histology and these powerful instruments, and we collect the observations. And what Tycho did was collected these observations, compiled them in the Rudolphine tables. And because these observations were very accurate and trusted and collected in the Rudolphine tables, this enabled Johannes Kepler to later come sit down with the data in the tables and discover the ellipse and change the way we view our universe. And the desire to explore and discover, these are the basic desires of all of us, and especially as scientists. And software is enabling us to take this to another level. And so how does it happen? Well, number one, we start with the instrumentation that creates the imagery. And nowadays, as I said, we're seeing smaller and smaller objects, more and more resolution, more complete views of the material that we're interested in studying. And we're seeing data coming from many, many different places. So in microscopy, we see data coming from light sheet, confocal, two-photon, live cell imaging, structured illumination, super resolution microscopy, and electron microscopy. And now we're bathed in all of these gorgeous images, and we need to extract the information from the images. And we're really set up in science now, and we have been for a little while, to really scale up our imaging and image analysis efforts. And by scaling up, some really special things happen. So one is, let's think about the case where we have a very high quality histology, really advanced cell targeting and imaging instrumentation. And now in this case, we're able to see the morphology of all the cells inside of a brain. Well, nowadays, our imaging systems are so fast and produce much data very, very quickly at very high resolution. We're able to do this kind of imaging at scale. So now we're able to look across many, many organisms and do comparisons. And by doing these comparisons, we start to gain new insights. So one is we start to find different cell types. We're able to categorize all of these things we see. We start to see statistical phenomena, things that are happening some percentage of the time. And we're able to do correlations between something like behavior and anatomy. Also, occasionally we're finding these needles in a haystack. So as we're imaging thousands, tens of thousands, hundreds of thousands of samples, we're seeing very, very rare phenomena, very, very rare and interesting phenomena. Then also, even within one organism, within one tissue, we're able to understand the relationships and the connections between the cells at a resolution that just was not possible before. And then finally, we're able to look at these images over time and begin to assemble how these organisms develop, how these cells move through time. So let's have a look at some of the basic problems of image analysis, and we'll start to see how image analysis can help us to extract the information from the images that we need. So let's look at, so here's a very basic detection scheme. So our imaging instrumentation works basically like this. We have something like pixels. So in this case, we have 9 pixels, and these pixels are capable of detecting some event in the real world. And we have the case of this worldly glowing object. And if we place this worldly glowing object in front of this array of pixels, it will trigger some reaction in this pixel in the middle. And now we can represent this event in the computer. So now we can digitize what we're seeing in the world. And this is very, very powerful and very, very special. So now we're able to store and hold on to the data, apply powerful mathematical tools to the data, come back to the data at a later time, and ponder it at our convenience. But we have a problem. We represent this event as ones and zeros inside of a machine, and because our imaging instrumentation is getting better and better, now we're able to collect more and more pixels. So now we collect arrays of pixels, very large arrays of pixels. And in addition, these imaging instruments are very good at detecting very subtle changes in the intensity of these objects, which means typically they have like a dynamic range that's better than the range of our eyes. Our eyes are very good at distinguishing many, many colors, but our eyes are not as good as these instruments at detecting these very subtle differences in intensity. And so one of the basic problems we have is how do we take a data set that's so rich in terms of the intensity value and examine it easily? And then another basic problem, and probably, yeah, all of us are dealing with this nowadays, and probably why you attend this seminar, is the 3D problem. So, oh, sorry about that. We have the 3D problem. So now not only do we collect very, very large arrays of pixels, 2-dimensional arrays, now we collect really large 3-dimensional arrays. And of course, we like to now collect 3-dimensional arrays over time. So how do we deal with all of this information? How do we explore it and extract information from it? And this is, of course, what software can help us with. And I think probably one of the core, really the core issue with image analysis is how do we take what we see with our own eyes and in our own mind, and how do we mathematically encode this in the machine so that we can share these procedures with other people. So let's look at this example. So here's an example of where we have we have an image. It's quite small, and the pixels have been scaled so that we can see some objects in the image, and these happen to be objects of interest. And if we do something like, say we're interested in understanding the volume, the size of these objects, we can do something very simple like use an intensity threshold to define a border around the objects, which in some cases this is completely fine, but in this case it's not completely fine, because if we study the image very carefully, what the experienced person knows is there are really 3 objects here that need to be studied. And the question is, how do we capture these 3 individual objects and measure their properties? And for that, we need something more sophisticated than a simple intensity threshold. So let's discuss some concepts of solutions. So how can software help us to overcome some of the challenges? So one thing is, now we have lots and lots of data, right? We have lots of pixels and we have lots of files. And one thing that software can do for us is it can simply provide a file and data structure that makes sense. So the images can be stored as a file. The results that we compute on the image, say these boundaries around the objects, can be stored as files. Settings and shortcuts and the analysis recipes can also be stored as files. And these things, we would like them to be in our control. We would like to know where they are, and we'd like them to be associated in a way that makes sense to us. And this is no small I think this is no small issue to discuss, because I think nowadays a lot of us, we're using our cell phone. We have these apps. The apps just are doing things, and we don't really know what they're doing. There's files being, going to the cloud, going locally, and we don't really know much about the files. But in science, we really need to know, where do the data live? Because we need to come back to it, and we need to potentially perform algorithms that depend on where these things sit, on what kind of hardware they sit, and so we want to have control over where the files are. Two is that we need a multi-scheme exploration of the images. So as we saw, the imaging instruments can have dynamic range greater than our eyes. So at least in the software, we want to be able to examine these various intensity values. We also want to be able to move through the planes seamlessly. We'd very much like to represent these arrays of pixels as volumes and make them appear as they appear in the real world. And we'll come back to that later. We also want to be able to look through these images through time. And in addition, we would like to be able to apply colors and various enhancements to the data sets. And of course, as we get results, as we capture the objects in the images, we want to be able to see those results really right on the voxels themselves. Thirdly, in some cases, a very simple analysis algorithm can work. So something like an intensity threshold can really give us the border of the object that we're interested in. But this is not always the case. And I think what we need is we need the ability to combine various operators together in customized ways in order to extract what we need from an image. And so we need a very flexible analysis tools. And finally, we want to have, I think it's very important that we have worldly experiences with our images. So we want to move from the ones and the zeros, and we want to take these high resolution data, and we want to give them back to our brain sort of in the way that we're used to dealing with it. So as you're prepping the tissue in the lab, you're working with the organism on a day-to-day basis, and you're familiar with, you're used to seeing it a certain way, and perhaps you want to understand the relationships of the objects that exist inside of the tissue. And so we want to be able to reconstruct it in a real world way. And this, I think, provides us with more information about the image and can really help us to start to decipher the three-dimensional spatial relationships between the objects and the images. And here's just a quick example of, here's a bunch of 2D slices. And here's what happens when we begin to make various kinds of projections of these 2D slices. So we take the many, many slices and we start to build something that makes sense with us. And we have lots of different techniques to provide ever more information about the relationships between the objects. And I think I really think this is an important topic, and I hope that we could talk about this in a future webinar. And then with the visualization techniques, we can do things like really open up an image. So in this case, we have some neurons, and they're labeled with a fluorescent dye, and they have a solid appearance. But using some direct volume rendering tricks, we can render the raw data in a way that really opens the image and enables us to go inside of it and explore it and really begin to see and feel the relationships, the three-dimensional relationships between the objects. And of course, this has a wow effect, and I think we all, many of us appreciate that, and we like to have maybe get the the picture on the cover of the journal. But I think this goes beyond the wow effect. I think what we do here is we provide more information in a single shot. So let's really look at some solutions. Okay, so here's what software is really going to do for us, and I'm going to walk you through a couple of different examples. So here are a couple of images, and we're going to do some different things with these images. So as I'm sure you can all see, we have some objects here. So clearly we have some bright pixels here, and we have some dim pixels here. And again, we have some bright object and we have some dim object. And the experienced mind can see, hey, we can see what is there and may have an idea of exactly what they want to extract from the image. But here's the issue, and I think many of you run into this, and it reminds me of the quote from Tolstoy's famous novel Anna Karenina: "All happy families are alike. Each unhappy family is unhappy in its own way." I think this applies exactly to images. So I think the ones that are happy and easy to analyze mathematically and algorithmically and at scale, they're all alike. They don't have optical artifacts. They have extremely little noise. There's perfect contrast around all the objects of interest. And each other kind of image, each challenging image is challenging completely in its own way. And I think we all run into this problem from time to time, where we sit down with our image and we have a concept of how we may want to grab this object or grab this object, and we start to apply algorithmic approaches, and they fail for some reason because there's some optical problem, there's some staining artifact or something like that. And I think we need to have a very flexible solution to do this. But the first thing we need to do is we need to be able to move through these images and do what I call scouting for the truth. So we have to be able to move through the planes, move through the volumes, and find these regions of truth, these objects that we know they really exist and we really want to grab them in a certain way. And then we also have to be able to find the problems. And once we find the truthful objects and we find the problematic objects, then we can really build an analysis paradigm for that image. So let's have a quick look at some of the paradigms. So one is, we'll start very simple. So we have, of course, the image with these two little, they're blobs for now. What do you call them? Blobs. And we can scale this image in a couple of ways. And if we scale it so that these appear quite bright, we can see that there's quite a bit of, there's bright pixels. But we know, at least I know, and I think probably a majority or all of you know, that there's nothing here. There's probably nothing here. This is real and this is not. And if we try to do something simple like use the intensity-based threshold to grab these objects, what happens? Well, we get the objects, but we get lots of other things that we don't want. And this is really a problem, okay? So this is especially a problem for when the images are really, really big, because what happens now is we have to devote resources. We have to devote our computing resources to dealing with all of these objects. Of course, we can later on do something like filter out all the small objects and keep the big objects, but what does it mean? It means we have to do some computation. So how could we skip this? How could we apply a simple filter to the image and solve the problem? So here's a simple idea, and I think probably a lot of you do something like this. So we can take the raw data, and we can compute the mean. So we can within a radius of 1 pixel for every pixel in the image, we can compute the mean value, and we write it back onto the into the computer, and then we grab the borders of the objects, and what happens? Well, things become very smooth. We lose all of these non-interesting objects, and we keep these objects. So I think this is probably something that's filtering is something people in image analysis do quite commonly. Here's something so this is also filtering, and then we'll move on to maybe a bit more of an advanced something a bit more advanced. So here's a case where we have objects in a cell. And these objects are associated with the cytoskeleton of the cell. And we may want to do something like grab the cell, because we want to begin to compute the number of things in the cell. So we want to understand cell by cell phenomena. And what happens is, if we try to do something like an intensity threshold, we kind of start to get the border of the cell based on sort of this out of plane light here. And we also get these sort of, lots of background objects. And so I show you here, we're getting background objects. We kind of get the border, what is roughly the border of the cell. And this may be good enough in some cases, but in some cases, maybe it's not. So what happens if we reduce the intensity threshold? Well, if we come in with a lower intensity threshold, we get less of these objects outside the cell. But because this staining is very like punctated, then we start to see these like openings in the cell, and that prevents us from really being able to grab the cell's morphology based on this punctate staining. You might be asking yourself, well, why would you try to grab the cell based on, you know, a stain like this? And the reality is we just don't always have the luxury of using exactly the histology that we want. And a lot of times we're looking at the data later on and we're thinking, oh, I have a great idea. If I can grab this cell, then I can do some kind of cell-by-cell analysis. And you're sort of thinking after the fact, what can I do to get the most out of the image, right? So, and here's a case where we've reduced the intensity threshold quite a bit, and now the cell is completely broken. So what I do in cases like this is, well, there's a couple things we can try. So one is we can try a different filtering idea. So we could try something like median, very aggressive median filtering. And what would happen in that case is the image becomes a bit more smooth, and we begin to be able to grab the outline of the cell. But there's a couple of things. It's approximately correct, but it can be quite time consuming, because in this case we have to compute this with a very large radius, and this takes up a lot of computational time. And when I see cases like this, I start to think about motifs. And motifs for me these are just little combinations of operations that work in certain cases. And I'm still learning motifs. You know, every week I sit down with a customer and we're discovering some new analysis motif that we can apply to other images. And here's a case where I would use quite a simple motif. So one thing I would do is I would take the image and first of all do an intensity threshold. So binarize it. So say only the pixels above a certain value are going to exist and have a value, and all the pixels that are below the value, now they completely become zero. And then what I would do is I would close these up. So I would basically do an operation that dilates these puncta, and where they join each other, they stay joined after an erosion. And now what we get is something, yeah, it's starting to look more accurate. We're following the contour of the real thing that we see in the image. And that's what's really important, right? We don't want to generate some kind of fake result. We want the borders around the objects to really snug up, hug real objects in the image. And so this morphology motif, which is basically, it's a mixture of opening and closing. It preserves clusters, so it keeps all of these ones that are very close together. They're preserved, but these little loners disappear. Very, very powerful motif. And then finally, we want to do things like we want to grab the objects and we want to study the objects. And sometimes grabbing the objects is not so easy. So in this case, we have two nuclei, and they're butted right up against each other. And we need something a little bit more advanced to grab these as individual objects. And so in this case, we might do something like a watershed to split the two objects. And so we model their morphology by doing a bit of a blur. We see the objects, and then we put a watershed between them. And then finally, we want to do something like understand their distributions. So how many objects are in this cell? How many objects in these cells? So let's before we really look at how the software works, this is how I see analysis. So I don't really see analysis as a series of steps. I see analysis as a series of cycles. So that means we sit down with our image and we scout around. We find these regions of truth or happiness, and we find these other regions of untruth or unhappiness. And we come up with ways to filter, denoise, and apply motifs. And these help us get objects of interest. And then we might sort, sift, and examine the objects of interest. And then if our images are really, really large, we have many, many images, we want to move on to other portions of the image. And that means we're going to apply this cycle to a different portion of the image, and we may find some other regions where this fails. And so we want to cycle through again, and we want to build analysis pipelines that work not just for 1 or 2 or 3 regions, but we want to optimize so that we get the most reliable, high quality results from our images. So I want to take you all into the real software environment and show you how you might do these things in practice. So I'm going to use Vision 4D for this. And what we'll do is we'll look at a simple example, and then we'll move on to this motifs example. So here's a very simple example. So let's just have a quick look. This is the obvious stuff. You want to be able to, of course, scan through the planes, scan through time, toggle things off and on, right? So we might want to toggle a channel off that we're not particularly interested in working on at the time. We might want to change the colors. And these things, you want to be able to do them very, very smoothly, very, very intuitively. And then that's what we can do in this environment. And now we look at this image and we know that it has some sort of this like salt and pepper, and we want to remove it. And how do we remove it? Well, what we can do is we can begin to apply various filters. How the heck would you know what filter to apply ahead of time? So there's 2 ways. One is you can go to the literature, study, talk to your colleagues, and really begin to make a real study of image analysis and become an expert at image analysis. If you're like me, I haven't spent my life doing image analysis. So for me, I just need to try things and see what works. This is exactly how I work with an image like this. So I would open up some filtering tools. I might know I want to try denoising, and then I know that lots of people can compute Gaussian blurs very, very, very frequently, and I might compute a Gaussian blur and just have a look and see what does it do. Here's what we see after, here's what we see before, and what I see is it's not removing this salt and pepper noise for me. And so I might just want to try something different, not even knowing what a mean or a median or a Gaussian does. I can begin to work with this image, and now I can see I'm preserving the border of the thing that I'm interested in, and I've really reduced significantly the stuff that I'm not interested in. And now I can come in and just touch on the image and start to grab various objects. And I can parameterize an intensity threshold interactively. And I think this interaction with the imagery is really, really important to the cycle of optimizing analysis pipelines. So let's have a look at another example. This is pretty, I like this one a lot. So we have this absolutely gorgeous image here. We've got multiple color channels, and we can explore through the planes, and we see it's absolutely beautiful. We see there's some chromatin material is labeled in blue. We see something labeled in red, which I would presume is something associated with the cytoskeleton. We see a bunch of little green dots. These are associated with proteins on the chromatin that are involved in the splitting of the cells. And let's just have a quick exploration of the image. So one way to explore it is, of course, to move through the planes, but another way is to really look at it in 3D. So we say, okay, let's enable a 3-dimensional view of the image, and now we can control the opacities and the transparencies of the various color channels. If we want to, we can toggle channels off. So maybe we only want to look at the chromatin. Perhaps we want to look at the entire sort of cellular morphology. Perhaps we want to really sort of begin to open up the image a little bit and be able to sort of see through condensed chromosomes. So we can begin to explore the image in the volume space. And for me, this gives me ideas about how I want to attack the analysis of this image. So let me give you a couple of examples of how we might analyze an image like this. So in this software environment, I have access to any portion of the image at any time I want. And one thing I did was I went and took, for the purposes of the webinar, I picked a cell. I thought, okay, I think it would be a good idea to show you guys how we apply these morphology motifs. So I just grabbed a small portion of one of the cells, and so I could really walk you through how I would apply and parameterize this morphology motif that we talked about before. And what I did was I actually stored the operation itself in this pipeline tool, and I'm going to walk you through it, but I'll walk you through it in sort of the way that, hopefully in like a natural way. I don't want to just press a magic button and show you, hey, look, it magically works. I want to show you how I would really work with something like this. So I would typically, I would focus on a region of the image like this, where I clearly see what I want to see. I want this border. I want to get it. And I have an idea about how to get it. And I would begin to parameterize an intensity threshold. And I would really come in and do this interactively. I would try different things. I would try low intensity values. I would try high values. Try like 2000. Probably the whole thing would disappear. Let's see. Yeah, we have a little bit left. And I would zero in iteratively on the one that I think is gonna work for me. And then when I'm happy with it, I would compute it. And then I would move on to the next step. And the next step in this case would be to start to close up, to sort of button up all of these open portions of the image. And again, we can do the same thing. We can close, very, very lightly. We could close very, very aggressively, like so. And the more aggressively we close, of course, the more computational time it takes. But this is how I would begin to build this idea of closing up the borders of the cell. And then finally, I like to do the, not finally, but the next step is I would do some opening, and I would try to remove all of these tiny objects. And as you can see with the opening set with this radius of 2, I can simply remove the objects. So you might ask yourself, well, how would you find all of these? Well, they're all organized in the menu here. So I can try different ideas via drag and drop. So I drag in denoising filters, I drag in morphology filters, and I drag them out. So they just go in and out, and I parameterize very, very quickly. And then finally, I might come to the point where I've opened it up, I've removed the tiny objects, I've got a pretty nice border of the cell, and then finally, I might want to do something like fill the hole. So then finally, we fill the hole, and I might want to do one last round. So I love to optimize. So an environment like this is really, I love this kind of environment because I can continue to optimize and add more operators. So if you look at my pipeline, at the core, it's something like, closing, opening and closing, but I add extra operators to refine, because I want to get as close to truth as possible. I just don't want a fake result. And so what I get is something, I'm starting to approach something that I would, something approximately true, or truer than I would get with something like a simple denoising. So let me move on to another example. So this is the watershed example. So let me, I'm going to find my little pipeline and show you guys how I did this one. So we go to this guy. And so here are here's a tiny portion of those green objects in the cell. And so here we can move through the planes. And of course, if we want to examine it as a volume, we can examine it as a volume. And in fact, let's just go ahead and do that. So what I'm going to do is I'm going to do something a little bit, let's say fancy. And I'm going to do a split view, so I can have the raw data rendered in this window, and I've got the 2-dimensional result rendered in this window. And just as a note, I think it's very important for me. I like to parameterize all of my analysis pipelines with a 2D view because I want to see the raw pixel. I don't want to see the rendered pixel. I really want to see its raw value. And what we do in this case is something pretty simple. So let's find, there is a case in this sample that I want to show you. So this is the case here. It's a case where you've got 2 hot blobs, and you want to grab them as individual objects. And this is where something like a watershed comes in really, really quite handy. So you could do something like drop in an intensity threshold, and you could try to parameterize it like this. I hope you guys can see that, where you sort of, you make it very restrictive, so you just grab the core of the objects. But what happens in this case is, as you look through more and more of the image, you're losing objects of interest, and you don't want to do that. So what happens is that after you've tried this, this idea and you find that it doesn't work, you say, aha, I can try something like watershed. And so we have an operator in Vision 4D called the blob finder, which does exactly what it says. It's very good at finding the blobs. So it finds these hot spots and it splits them. And then what I can do is compute it. And then once I compute it, aha, let's do something a little bit more fancy. Let's see if we can, show the annotations. Well, let me first show you the annotations here. So, boom, boom, boom. All right, there they go. So now we've created real objects, okay? And let's say we're interested to see where is this object in 3D. So what I'm going to do is I'm going to link these to the two viewers so that when I click on an object over here, so I'm going to find that little split. So if I click on an object here, I should see that object highlighted over here. And let's make it a little easier for us to see what we see. So let's go ahead and maybe put a bounding box around it. And so here is this object that we see over here. And here, let's click on this one, is this object over here. And so by parameterizing this operator and viewing it in the 2D mode and the 3D mode, we can begin to approach an ever more truthful representation of all of these blobs in the image. So let's move to another example. So let's go back to the whole thing. Oh, or to the, well, not the whole thing. Let's go back to this guy. So this cell. So now we're really looking at the cell. Okay, we're going to look at it in 3 dimensions. And what I've done is I've already computed. Let's go back. I need to go backwards. Okay, and we're going to just move over back to the cell. And what I've done is I've already computed a bunch of stuff. So I've computed the border. I've used this little motif to compute the border of the cell. And I also used a similar motif to compute the border of all the chromatin in the cell. And I found something pretty interesting. So I found that there's actually 2 groups of chromatin in the cell. So there's sort of this large group and there's this small group. And by using these analysis tools, what I was able to do is find the small group all by itself and find the large group all by itself. And what does this set us up to do? Well, it sets us up to do something like we could count these centromere associated objects inside of here. So let's just do this. Let's do this quickly. So what we could do is we could take, let's take this small chromatin object, and let's name this the small chromatin object. And then what we could do is give the pipeline all of those bright green objects. So we have lots and lots of bright green objects. There's maybe 5,000 of these bright green objects in the image, and we could do something like object-based co-localization. So we could take this small chromatin object, and we could use it as a reference. So we take the small one, we use it as a reference, and then we could take these green objects as a subject set. And we could say we want to report all of the, say, objects, all the green objects that are completely covered inside of the chromatin object. And so that's exactly what we get. And so now we can go to our 3D view. And now what we've done is we've pulled the chromatin, or these centromere associated objects that are inside the small group. So let's leave the environment. And let's sort of, review a little bit about it. So in the environment, what we have is a completely modular design. So we have modules for bringing the files in. I didn't show you this part because it's pretty boring. It's you drag the file onto the desktop and it's in the environment. But we did start to look at, of course, the viewers. So we're able to view the data in the 2D mode. We're able to view it in the 3D mode. We're able to view our results in a table. And we have controls. So we can turn the image. We can move through the planes. We can use a little dropper tool to parameterize our intensity thresholds. We have a results database that's computing features of the results for us that we can utilize. We didn't talk about transformations, but these are quite powerful. So transformations and corrections like drift correction and bleach correction, all of these sort of things exist in a module. We saw this. So these are all of our analysis tools. So this is a whole multitude of operations, filters, morphology, previews. It's completely logical. You saw how I renamed some of the operators for small chromatin or large chromatin. And then finally, we have a storyboarding tool, which enables us to create the movies. But the thing about this modular design is that we can really customize and move easily. And I think that this is, at least for me, the most important thing when it comes to image analysis. I need to be able to move through the images and move through different operations to figure out what the answer is going to be, because it's just not always obvious from the raw data how you're going to get to the final answer. So nowadays, I already mentioned, these imaging instruments are producing ever more data, more resolution, more time, not only in X, Y, and Z, but also in time. And so the amount of data is really exploding. And so what does it mean? It means the size of the data sets are bigger than the size of the computer. So I think nowadays we're working on workstations that have something like 500 gigabytes of RAM or 1 terabyte of RAM. But what do we see routinely? Images that are 10 terabytes and above. So these images are far bigger than the size of the computer. And what happens when you use your computer to hold all of your data? Your computer can become unstable. And so what we want to do is we sought to address the gap between your processing needs and the computer size. And so what Arevis did, which is not unique to Arevis, but I think the way that we do it is pretty special, is that we create a redundancy-free data structure, and it makes efficient use of the system resources. So we don't have to touch the RAM, okay? So your computer can remain stable, and then we can give information to RAM when we need to and when we want to. And the Arevis file format, which is called SIS, gives you access to any portion of the image at any resolution, at any point of time, any region of interest inside the image for any kind of operation. So we can take any little 3-dimensional, 2-dimensional, 3D over time portion of the image and give it to various operators and very rapidly build analysis pipelines and workflows. Very, very important for large data, because the last thing that any of us want to do is waste our time. We do not want to try to compute via some approach that's going to fail. We want to know that it's going to work. And so SIS gives you the ability to check, to make sure that, to number one, figure out what's your idea, what's your analysis idea, and then secondly, is it really going to work? Is it worth to apply to the whole image? So I think about, if I take a big step back, and I look at, okay, what are all the resources that we need in this day and age to investigate really big data sets? And a lot of you are probably here because you have this problem. You have ginormous images and you need to be able to work with them efficiently. So one thing is, of course, minimal obstacles. And what does that mean? It means, well, you want to work on any hardware. You don't want to have to go somewhere where you have to wait for a special machine or special hardware. So you really like to work from anywhere. And I'll tell you right now, I'm sitting at a laptop computer. I work 99% of the time from a laptop computer. Even with absolutely ginormous images, I will put the data on a drive and plug it into the laptop computer. So out of convenience. And I'll work on an airplane. Okay, so you want to be able to work anywhere you want, in your lab, at home, on a workstation that's very, very powerful. Anywhere you want to go, you want to be able to go there. You want stable access to the data, so you don't want to crash. And this is one of the things that when I'm out interacting with our customers, I find this to be one of the things that really irritates them when they, not about our software, by the way, it's just in general something that they don't want to deal with, and that's why they come to us. Okay, they invest lots of time in working on their images and building some kind of analysis pipeline or paradigm, and then they're having problems with software stability. And so they come to us and they say, hey, can you help me with this? Because it's really frustrating me. And so what we endeavor to do at Revis is build a very stable platform. And one of the reasons why it's stable is because it's not going to eat your entire computer. Okay, but there are other reasons too. But it's quite a stable platform. You also want to be able to work with the data immediately. You don't want to be locked out of the software. And this is one of the things that our developers take very seriously. They try to build the software in a way that enables you to not only work as soon as the data is there, but also to continue to interact with it while you're running various operations. So typically, you might start on a little operation, but you have another idea, and you want to try that idea. And so Vision 4D software, we try not to lock you out of the software. We want to enable you to express yourself on your images and continue to think even while your computer is thinking. And then flow of information is extremely important. You want a rich visualization of the data, and you want to see the results right on the data volumes. Rich visualization means options for making this object look real based on the raw data. Take the raw data and give you information so you can see what you need to see, and we want to see your results right on the data. We also want to give you natural experiences with the 3D data. This means not only natural controls with your keyboard and your mouse, but it also means natural, using the latest technology like VR technology to let you reach into an image and touch it and interact with it. And we'll talk a little bit about that. We also want you to be able to publish your results, and we want you to take the beautiful images that you've created and make a spectacular image. And by spectacular, I mean information rich. You want to put a panel in the paper or on the cover of the journal that conveys as much information as possible to the readers. Also, we spend quite a bit of time, and we're very interested in building web interfaces and collaborative workflows. So we also have tools, which we didn't look at today. I'll tell you a little bit about it, but we have tools for enabling you to interact with these SIS files completely via the web and in a completely collaborative way. And I think we all agree that that's really important in this day and age, and it's going to become ever more important. And then finally, we have this very flexible computational tool set, and we really just started to scratch the surface of that today. But in my view, the important thing is that you need to be able to do rapid prototyping. And rapid means, it means that you got to click, you want to be able to click around and get your ideas into the computer without writing lines of code. And I think that although you can, if you want to in a Revis, extend a Revis by doing some more advanced things like writing code, for the most part, you can navigate the Revis environment in a very easy way with a mouse and the keyboard. So we at Revis, we're endeavoring to do much more than just this standard experience with the data where you sit down at the computer and you sort of move through the planes and you parameterize a pipeline. We really want you to be able to move through the data in a way that it's very easy. You're seeing everything you need to see. It's very interactive and sort of a continuous flow of experience. And we want to connect you with your collaborators, with other people. We want you to be able to share your results very easily. And we want you to be able to work together. And so the way we endeavor to do this is, well, we're trying to build future-facing tools. So we imagine that you could store your data anywhere you want. You can have it, you could store it on the cloud, you could store it on a local server, you can store it on your local machine. And then what we enable you to do is, through our environment, access it via desktop devices. You can even do it on mobile devices. don't tell our developers, but I really do that. Sometimes I access the data that we hold on the web in the browser on my telephone. And then also you might want to plug in and use the latest virtual reality tools to analyze the data. And I'm going to just very, very briefly just give you a little taste of what we have coming in VR and in the collaborative workflows. And then we'll wrap it up. But I think this stuff is pretty exciting, and I hope that it's a topic for future webinars. So in the VR space, what you're able to do is really immerse yourself inside the data, and you really start to do something different. Your brain starts to see more. You start to feel it as an environment, and when you can reach out and touch data, you can do things very, very quickly just by touching it. And I wanted to show you guys a really quick example. So you can start to solve really hard problems. So this is kind of a hard problem. So when we get FibSem data, and our customers want to see certain objects inside of FibSem, I think a lot of people would naturally want to do something like machine learning AI and try to train a machine to find it. But it turns out that if you ahead of time, oh, sorry about that. Oh, sorry again. Let me play this. With our VR technology, which we call InViewer, what you can do is you can hold that FibSem volume out in front of you, and you can really slice it up any way you want to. And then what you can do, which is, I think, a very, very powerful motif. So again, this is like a motif, but a more advanced kind of motif, is what you can do is you can sculpt over the regions where you know there's something you want. So it's sort of like you can target your AI onto certain regions, and that makes the problem so much easier for machine learning. So what we do a lot of times is take the volumes, we reach out into the volume, we sculpt right on it, and depending upon how big the volume is, it might be a matter of minutes, it might be a matter of an hour or two, but in the end, after you've sculpted it, then you train your AI on the volume and you get something really nice. Like in this case, we grab all the collagen. So this is really fun. And then just very briefly, we also have a product called the Web View. And the Web View is a way for you to interact with the SIS files and the results completely via the web. And if you want to, in a completely collaborative way. So multiple people access the data, and you guys can you can apply analysis pipelines to the data. You can do manual, sort of like what people would call like crowdsourced kind of annotations. And then the beautiful thing is that all of these different tools, they all fit perfectly together. So they're all using the Arrivis SIS file format, and they all plug into each other. So that means that you could build a completely custom imaging workflow in your lab. So this is where we end. But there's one thing I want to share with everybody. So when we were preparing the webinar, we were talking about, okay, how do we introduce this idea of image analysis to everybody? And I said something like, image analysis is something we do every day. I just went to Whole Foods last night, and like there's a particular kind of whole milk that I like in Whole Foods. And so I'm doing it. I'm walking up to the freezer case or the refrigerator, and I'm picking the milk that I want. And so I thought, okay, how about I'll go to Whole Foods, which I did last Friday, and I took a picture of the freezer case in Whole Foods. And so here it is. And I used Vision 4D, and I used a bit of the trainable. So I have to admit, I used the trainable segmenter and to output sort of a probability image. So I trained it on what's a gallon versus what's not a gallon. So this is something we didn't really talk about today, but I did that. Plus I used exactly the morphology motifs that we talked about today, opening and closing, to sort of solidify those gallons and remove the background objects. And what I did was I trained Vision 4D to do some stuff for me. So I trained it first to find a high probability of, right, let's see. Oh, I have to show the annotations. So I trained it to find me objects that were a high probability of being a gallon. And so I found all my gallons. Then I found whole milks. So I always buy whole milk. So I had it find the whole milks. And then after I had it find the whole milks, I had it find the Whole Foods brand, because that's the brand I particularly like. And then the jug that I picked last Friday night was a jug that was in the front and didn't have a dent in it. And so I used the analysis pipeline to find the jug without the dent that was in the front. And I think this is yeah, it's kind of a funny, cute example, but I think this is really what we all want to do. We're looking at our images and we're the experts. We know what's in there. We know what we want to measure. We see the phenomenon. And now we need to capture it because we need to share it with other scientists. And that's exactly what I did here. I encoded the process that I went through in the Arevis environment. And that's exactly what you want to do with your images. So I think that's where we'll end it. So I want to thank you all for meeting with us today. It's really my pleasure to speak with you about this. And it's been my pleasure in working with Arevis for the last almost 3 years and meeting many of you and sitting down with you and working through your image analysis problems. I've learned a tremendous amount, and I hope that you have too. And I hope that we can continue to work together. And I'll just have to say, it's been a real pleasure to work with everybody at Arevis. Arevis really cares about delivering a product that's very, very powerful, that provides a very, very high quality of result, but also provides a joyful and high quality experience to you. And then finally, I'd like to thank Nick and Martin for working with us on developing the webinar today. And with that, I would be happy to take questions.
Thanks, Chris. That was an excellent and thoroughly enjoyable presentation. We have a few questions from the audience. If anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So I've got one here from Niraj, and they ask, in the 2D viewer, can you draw an outline around the object of interest?
Absolutely. So let's have a look. So I think it's always better to really show you how it happens. So let's have a look together. So we have a tool for drawing. So you can draw different sort of shapes. So you could draw a circle, square. I would say in most cases we're doing something like drawing a polyline around an object. So let's first simplify. Let's simplify everything for ourselves. So what I'll do is I'll hide a couple of channels, and I'll even make. I'm going to make this gray, so hopefully everything appears a little bit more clearly on your screen. So we have a couple ways of doing this, and the two ways that I do it the most is simply to draw a polyline. So I select a tool for adding an annotation object to the image, and I just will use this to simply draw the polyline around the object of interest. And I'm going to take a guess about what your next question would be. I would guess that you might ask, could we do this in 3D? And the answer is yes, and there are a couple different ways, but one of the ways that I would tend to do it is I might move to another plane, and I might draw another polyline on another plane in this way. And then what I can do is I can interpolate between the two polylines to create a three-dimensional object. And so I'd say that's a fairly typical way that we would outline objects. But there's another way. We could do something like magic wanding. So we have a magic wand tool, and I can parameterize the magic wand. I've got like a tolerance value that I can apply, and I can use that to also grab the objects in the 2D viewer.
Okay. Brilliant. Thanks, Chris. I've got a question here from Jan. Is the Arevis Vision 4D software compatible with marker-free technologies?
Can you repeat?
Is the Arevis Vision 4D software compatible with marker-free technologies?
Marker-free. I don't know. I got to admit, I don't know what marker-free is, and it's something I would. Maybe our developers or other application engineers are familiar with this kind of technology. Unfortunately, it's not something that I'm familiar with. So I have to kind of either you have to explain it a little bit to me, or somebody has to explain it, or I have to find out from somebody else.
Okay, that was the question I got. Okay, moving on to the next one. Garima asked a question during the live demo of the Arrivis Vision 4D. Is it possible to measure the orientation, coherency, and length of the tubulin?
So I would say no. I don't, and yeah, maybe we have to sort of get back to you on what we really can do. I would say definitely not directly. So what we would do is something like, let me show you. If we go to, if I go to this, so we're going to look at the, so we've got lots of objects. And the objects, we compute lots of different properties for the objects. So we compute things like their length, their aspect ratio, their position in X, Y, and Z. And I could imagine that we could so all of this information is exportable into the spreadsheet. So we can get all of these computed properties out of Vision 4D. And what I would guess is that in order to do this kind of thing, we would pick certain properties of or certain features of the objects, and then we would compute that stuff outside of Vision 4D, at least for now.
Okay. I have a question here from Paula. She just asked, is it compatible with the Zen files from Zeiss?
Yeah, so it is, and we support the import of numerous file formats. And when the files are coming from the big reputable imaging companies like Zeiss, usually these are cases where the import is the absolute easiest. So when the files are coming from Zen and from Zeiss, we're able to they share all the information with us about the file format, and then it makes it really easy for us to catch all of the relevant information from their files. So in those cases, in this case, it's really as simple as a drag and drop.
Okay. And question here from Kathleen. Have you done any analysis on non-fluorescent, non-SEM data, such as differential interference contrast microscopy?
I have personally not done. Oh, I have done. So I have recently done. So yeah, the answer is yes. And so we've looked at we've looked outside of fluorescent. So I want to say the tools in Vision 4D, it's very, very comfortable for working with fluorescence images. I think we make it pretty easy. With SEM, I kind of find it not too hard to work on the FibSem. I think there's certain tools you need for working with these blocks of EM data, but that I would like to see in the future in Vision 4D. But it's pretty good. I'm really having quite a bit of fun with that. With things like 3D X-ray data, it's a complete joy. So you're working with something like CT imagery or medical MRI imagery, and that kind of stuff I think is a complete joy to work in Vision 4D. The DIC stuff, it's a bit more challenging. So I just started to work with some of these with some time series earlier this year, collected in DIC. So there's like a wide field or bright field and DIC. And at first it was new for me, but I figured out the motifs basically. So I have some ideas, and I'd be happy to sit down and work with you in a web session and show you some of these motifs. But I tend to use the trainable segmentation. So a bit of machine learning and a bit of morphology and a bit of filtering in various motifs in order to catch the cells from the DIC images. And I would say that the pipelines that I had to build were a little bit complicated because the cells were reaching out and touching each other very, very frequently. And so there's just like it's like spaghetti noodles, all intertwined. And that makes things a little bit more challenging. But yeah, I was pretty happy with the results. So I would say, yes, we can work with DIC images, and I'd be happy to sit with you and look at some of the motifs.
Okay. I've got a question here from Oluwakimi. Actually, a problem where their they've seen artifacts that have been mistaken for metastases in their samples. Is it possible that 4D imaging can an analysis can help that problem? Is it possible to separate artifacts from metastases?
Yeah, it depends on, right? It depends on what's the nature of the artifact. So it's the kind of thing where we want to be able to explore an image like that very, very easily. And that's of course what you can do in a software like Vision 4D. And we would have to get a sense of, what is going on here. So sometimes we would have a case where the artifacts are, it's like an optical sectioning. It might be a problem with the clearing is not optimized, or the imaging parameters are not optimized. And so what you're seeing is sort of some out of plane optical issues. And there are some cases where we can account for that, and then focus just on the regions of what I consider to be truth. So I would say yes, it just depends on the images. In the simplest case, it may be that in some optical sections you have a problem, in other optical sections you do not, and you might just simply want to focus just on the optical sections where you don't have the problem, and that way you're not misled. But we should have a look together, and we can figure it out.
Okay. Next question here is from Baptiste, and they're asking, are there any quality control algorithms for the reconstruction of 3D SIM images?
Quality control algorithm for, so let's think, what do you want to measure? So I would say we don't have something like a quality control operation or something like that, but we could do something like build a pipeline that performs some series of operations, grabs some objects, and reports to you properties of those objects that you could use for quality control. I think one of the first things that I did a couple of years ago was I looked at data that was coming out of a very high throughput process. And I was interested to build a pipeline to measure histology artifacts, because I was curious about what's the performance of the histology over time. And so I did exactly, something like this. I built like an algorithm I built a pipeline that could grab certain artifacts out of the image, and by counting those in the various data sets, that gives me some idea of what's the quality over time. So maybe it would be something similar. It would be some kind of a pipeline that would extract a specific feature in your imagery, and then we would compute for you certain features that are important for quality control.
Right. Okay. Question here from Esther, and she asks, how does the Arrivis software interface with existing image capture software provided with the instrumentation?
So that's, I think we kind of answered that. So it's in some cases, like in the case of the Zeiss instruments, I think most of the time when I'm sitting with a Zeiss customer, I'm finding that the import is just really, really easy. They're saying, oh, this is a complex image, we've got lots of channels and this and that, and I just drag and drop it, and the whole thing is working perfectly fine. But the import tool in Arrivis Vision 4D, we didn't really I didn't talk about this, but the import tool itself, let me just try to fake it. I'm gonna just grab some random documents and try to fake for you, what might happen if you've just got some TIFFs coming off of, any old instrument. So let's say you built your own instrument and you're just making TIFFs, and we're Arrivis Vision 4D, you need to assemble them in a certain way. Vision 4D has a very flexible import tool, so it allows you to choose from common scenarios. So in this case, I just picked a couple of Z stacks, so they could be TIFFs, and then it's asking me, hey, should all these Z stacks be assembled as a bunch of planes on top of each other? Should they be assembled as time series? Does each one represent a color channel? And then if none of these scenarios are covering it, then I have a completely custom import. So I would say, like, I very rarely, if ever, I don't know if I've ever run into a case where I couldn't get the data from an imaging instrument into Arrivis. It's we really support the import of lots and lots of different files, and being able to import files like TIFFs and JPEGs and I mean, I imported that milk picture from my cell phone. So being able to import files from anywhere and be able to control how they're assembled, it means you can pretty much get the data into the Arevis environment from anywhere.
Okay, brilliant. Question here from Lindsay, and she asks, you talked about the capability to handle and visualize and analyze large data. What's the limit or the biggest image size you've had?
That's a great question. There is some kind of upper theoretical limit, and it was computed. It was told to me it's some weird name. It's not petabyte. It's like femto mega gigabyte. I have no idea what it is, but there's some upper limit. It really exists. But in practice, I personally have worked with data sets as big as 12 terabytes. And I will say that the performance of the software was excellent, stable. I was able to do the things I needed to do with those images. I will say I had multiple 12 terabyte images, open on my desktop at once. So probably the most amount of data I was accessing at one time was maybe something like 30 plus terabytes. And the software was really stable, and I'm able to focus in on portions of the image, parameterize pipelines. We've done quite a bit of movie rendering and screenshot rendering from these really big images. So let's say for me, it's something around 12 terabytes in a single image. It's maybe over 30 terabytes in a session. And I think our development team in Rostock has had their hands on a single image that was a bit bigger. It might be on the order of 15 or 16 terabytes.
Okay, brilliant. And question here from Laura, and she asks, can you also handle time-lapse images to do something like tracking over time in 3D? And can you also measure intensities or other features along a track over time?
Yeah, absolutely. So that's a great question. And I think we had a little bit of debate about whether we should cover the tracking stuff in this webinar, and because we all like it very much. It's really cool, but we thought it might be a little bit too much for the webinar. But we can absolutely do tracking in 3D. Maybe I can make Vision 4D track for us quickly here. I have to say that Vision 4D is really good at working with time series. This is a real strength of the software, and we should really do a webinar that's specifically about the time series in the future. So this one is a time series, and so we got all these objects waggling around in here. Let's see if maybe I can compute something for you quickly. This might be a little bit time consuming to compute this mean filter, but I compute it anyways. So there's a little progress indicator here, and then boom, I got all of these little, hopefully I get all these little red objects. Let's see. We'll see. Okay, so I got a bunch of objects, and then I'm going to do a tracking operator. So we have an operator that I can insert into the pipeline, and I can parameterize it. And now what it's done is it's tracked the movement of these objects in 3D. So if I flip over to our 3D viewer, there's a bunch of, maybe like not like ideal tracks, because please, I'm just doing this like on the fly. Okay, so please forgive me there. And I'm going to change the lighting so that it's a bit easier for you to see, hopefully, the objects. So I'm going to add some shadows and some. So this red object, now you can see we have its position, its track computed through time. And so what we can do is let's go to the table. There's all kinds of cool things I would love to show you about Vision 4D. That it's hard to, can't really show it to you in a 1 hour webinar, but now I get to show you some stuff. So this is kind of fun. So here's the track. So I can click right on this image and interact with the let's store these. Why not? So I'm gonna take it all the way. So I'm gonna complete. Whoa! All right, we got lots of segment surfaces. Let's just view them as centroids for now. And let's catch this track. So I'm going to really click on that track, and I'm going to group all of these. I'm going to group all of the results by the track. So this object lives on a track. It's called, in this case, track number 3. And the red object is represented in time by all of these segments. And so what I can do is I can go over to what we call track timeline view. And we could do something like, let's look at its volume over time, something like this. So for this object, now I'm computing its volume over time. And maybe the reason why it's decreasing is because I didn't photo bleach correct the image or something like that. So there's probably a reason for it. But my point is that you can absolutely look at the property, track the objects through time, and access their properties through time.
Okay. This question from Kieran kind of follows on from that, and you said it's possible to export videos of your 3D data. So how customizable is it?
Quite. So again, this is something that I could do an hour. We could easily do a webinar on the video export stuff. So, and it's something that I personally find really fun. So if you like to tell stories about the data in the movies, Vision 4D is a really good tool for that. So let me show you what the interface looks like briefly. So I'm gonna get rid of the spindle, this data set for a second, and we're gonna go just let's focus on this one. So if we take our little cell of interest, let's put all of our colors back on, and let's make this blue again. And then let's look at it in 3D. And let's turn off all of the segments, and let's change the lighting. So as you can see, there's lots and lots of controls, and I know where they all are. So it's easy for me to go around and click on all of this stuff. So I'm not trying to make it look easy. It's just I would say it's completely discoverable. Once you know where all this stuff is, then it really becomes easy. So let's have a look at the storyboarding tool. So we have a storyboard. Oh, let's see what this looks like. Oh, I like that. I like that a little better. Okay, so let's look at the storyboard. So we have this storyboard tool, and it works like you assign keyframes, and then what Vision 4D does is it interpolates between the keyframes. So let's say we want to have this sort of angle on top view as a keyframe, and then let's say we want to do something like let's barrel roll our way into the cell. Okay, like everybody at Revis is laughing right now because I always am doing something crazy like barrel rolling my way in, like Star Wars style, into the cells. Okay, so let's say we want to do something like that. So we want this to be the first keyframe, and I just double click on it, and I'm there. I'm really at the keyframe. It just loads up the visualization settings, and I click here, and I'm really here. What Vision 4D does, if I hit this play button, it does a interpolation between the first keyframe and the second. And then what Vision 4D enables you to do is control that transition. So you can control how long it's going to take. You can control whether the camera moves in a linear way, accelerated and decelerated way. And these are really great ways of focusing on objects and building tension in your movie and all this kind of stuff. You have complete control over, say we want to fade the red channel out. So I might want to add a keyframe where I faded the red channel. So now when I play the preview, I get the barrel roll, and then when we get close to the chromatin, then all of this red stuff is gonna fade away. And so I may want that fade to happen more quickly. So in that case, I would just go to the transition, and I might make it, instead of a 5-second transition, I can make it a 2-second transition. And then another thing that's, I would say, that makes storyboarding movies really easy in Vision 4D is that you can edit these keyframes really easily. So you see exactly what they are, and you might say, I really don't want to have the red channel showing in this one anymore. So what you could do is just double click on it, you reduce it, and you might even say, forget it, I don't want the blue channel either. So we take those away and we right click on it and we replace it. So in Vision 4D, you have this visual indicator of what your movie is going to look like. You have complete control over the transitions, and you can edit very easily the keyframes. I think it's a very logical and intuitive approach, and we get quite a bit of feedback from our customers about this being a really good feature inside of Vision 4D.
Okay. Well, that's all the questions we have for you, Chris, and that brings us to the end of the seminar. Thanks again, Chris. That was a fantastic presentation and a great discussion. And thanks also to our sponsor, Arrivis. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can also see the other webinars we've lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at Arrivis and Bitesize Bio.